Hello guys, and today we are going to start the next topic, which is the Renin Angiotensin Aldosterone System, and it's a very important mechanism, more of to say regulatory mechanism in maintaining the blood pressure and blood volume. So let's get started. First of all, renin-angiotensin aldosterone, or so-called RAAS, is important for maintaining the blood pressure and blood volume in case of hypertension or hypotension. Okay, for example, if a person is bleeding or is suffering from diarrhea or vomiting, his or her body fluids are going to be lost in different ways, right? So he is going to suffer from hypovolemia and hypotension, and he is losing the blood. So in this regard, how this system is going to be activated, first of all, it's very important to know that what are the organs that play a role in the RAS activation. First of all, the main organs are listed below. First is the central nervous system. It's the sympathetic outflow by the vasomotor center in the medulla oblongata. We will discuss it in detail. Kidney, nephrons, liver, the hepatocytes. Pulmonary circulation system, more specific, the endothelial cells of the pulmonary capillaries, and renal cortex, which is known as the adrenal cortex. To be more specific, the part is the zona glomerulosa. So, RAS activation overview. Let's take a scenario that if a person is bleeding, its blood pressure is going to decrease. He is going to suffer from hypovolemia because the bleeding is occurring. So, system response. When a person is bleeding, the amount of blood in our general circulation is going to decrease, and in response, the kidneys are going to be hypoperfused. What does it mean? It means that there will be lower blood flow to the kidney. It's known as the reduced renal perfusion, and in response to the reduced renal perfusion, the kidney will detect that something is wrong with the blood pressure, right? So it's going to release the enzyme known as renin, on which the renin-aldosterone system is based, right? So in basic concepts, there are three components of the renal corpuscle: afferent and efferent arteriole, and the glomerular capillaries. We are going to discuss about what are the pressure measuring devices in the kidneys that are going to assess the our blood pressure. In case of hypotension, how our nephrons know that the person is suffering from hypotension, or in case of hypertension, how our nephrons know that there is high blood pressure. There is a special type of modified smooth muscles in the afferent arteriole known as the juxtaglomerular cells, and they are responsible for measuring the blood flow to the nephrons, i.e., they are baroreceptors, like, right? They are pressure measuring or pressure sensitive devices. And to recall that both kidneys contain 2.4 million nephrons, and each of the nephron has its own pressure measuring system. It's known as the juxtaglomerular cells. Juxtaglomerular cells are nothing but specialized or modified smooth muscles of the afferent arteriole. Okay, and when there is blood flow reduction, when there is reduced renal perfusion, the pressure will decrease, and these cells that are sensitive to the pressure, they detect that pressure is low, and they are going to trigger the renin release. Okay, they are going to trigger the renin release. And in this regard, these cells have double action. First, they are responsible for assessing the blood flow, either the blood flow is more or less. And second, they are going to initiate the release of renin, and that's the enzyme we need for the hypotension conditions, right? When we are losing the blood or when we are losing the body fluids. And in other contexts, we can say that these cells work as endocrine glands. What is the definition of endocrine gland? Endocrine gland are those glands that secrete their products, that secrete hormones, and those hormones work on the distant parts of the body and they travel via the bloodstream. Okay, so in this regard, these cells are functioning as the endocrine gland as well. First renin is released in response to the pressure reduction, right? Baroreceptor function. And the second is most important, the sodium detection. Let's talk about it. First of all, as you know that when the blood is filtered in the glomerulus, all of the substances or molecules present in the blood are filtered except the plasma proteins and the substances bound to the plasma proteins. For example, water, electrolytes, glucose, amino acid, weight products, creatinine, urea, all of them are filtered freely. Okay, and most important thing is that this is the PCT, this is the loop of Henle, ascending part of the loop of Henle, and this is the distal tubule. At the junction of the distal tubule and the thick ascending part of loop of Henle, there is a specialized group of cells. These are modified cells. These are tall and dark under the electron microscope, and we just name them as the macula densa because they are looking as dark as compared to their surrounding cells. So these are nothing but modified cells. These are tall and dark, sensitive to the sodium. Okay, and this is known as the sodium sensing system. So first of all, we have discussed that there are juxtaglomerular cells. These are modified smooth muscle cells in the afferent arteriole, and these are responsible for the pressure detection, right? And the second is the macula densa, present at the junction of the thick ascending part of the loop of Henle and the distal convoluted tubule, and these are sodium sensing system. So first, the juxtaglomerular cells are baroreceptors and macula densa are chemoreceptors because they are detecting the sodium concentration in the tubular fluid. So let's see in this diagram. As you can see that this is afferent arteriole and this is efferent arteriole, and here is the glomerular tuft of capillaries. As we know, the juxtaglomerular cells are present in the smooth muscles of the afferent arteriole. This is the juxtaglomerular cells, and at the junction of thick ascending segment and the distal convoluted tubule, not shown here in this diagram, there are specialized cells. As you can see that this area is slightly more darker than the surrounding. This is known as macula densa. This is the sodium sensing system. And how these cells are placed in this system. First of all, there is the connective tissue that connect these cells, and this is the known as extraglomerular mesangial cells. Okay, this is extraglomerular mesangial cells, and in this mesangial cell there are two types of cells known as the lattice cells and the Pulkison cells. These are nothing but they hold the structures together and they are functioning as the connective tissue, right? And why we call this apparatus as the juxtaglomerular apparatus? Because this is located adjacent to the glomerulus. So what are the components of the juxtaglomerular apparatus? First is the juxtaglomerular cells, the smooth muscles, the modified smooth muscles of the afferent arteriole, and the second is the macula densa, the sodium sensing system in the distal part of the nephron. Coming back to our scenario that when the blood pressure was low, filtration is low, right? And the fluid moving through the tubules of the nephron is slow because the filtration is slow. Reabsorption will be more or less. Reabsorption will be more because the cells will have more time to capture the solutes present in the tubular fluid. So what is the concentration of sodium when it exits the proximal convoluted tubule? It will be decreased. There will be low level of sodium because a lot of sodium has been reabsorbed in response to the low glomerular filtration rate. And when this low level of sodium reaches the macula densa, as you know that macula densa is sodium sensing device or sodium sensing mechanism, low level of sodium triggers the macula densa to release two type of substances. First is the nitric oxide is a vasodilator, and prostaglandin, it's also afferent arteriole vasodilator. So these vasodilators are released in response to low GFR. Because there is low GFR, so kidney detect that the blood pressure is low, but To conserve or to maintain the GFR, to increase the GFR, we must need vasodilators of the afferent arteriole to increase the blood flow towards the nephron, and also in response to the low level of sodium reaching the macula densa, it triggers the juxtaglomerular cells to release the renin, and renin is the play of this system. So, in what context the renin has been released? Under two influences. First is the pressure change, the baroreceptor system, and the second is the sodium change, the macula densa system. Let's talk about the carotid system and renin. Not only the renin is released in kidney, but in response to other systems, the renin is also triggered by other systems. So first is the carotid system, second is the central nervous system. So let's discuss about it. Carotid system. Where is the carotid bodies located? Carotid bodies are located near to the carotid sinus. It's like a bulging point of the carotid arteries at the bifurcation of the common carotid artery. You know that common carotid artery bifurcates into internal and external carotid artery. At that same point, carotid bodies are located. And carotid bodies are blood pressure sensitive. In case of hypotension, when there is reduction of the blood flow towards the carotid bodies, these bodies are activated in response to hypotension, and they transmit signals to the central nervous system via glossopharyngeal nerve or the ninth cranial nerve. And when the glossopharyngeal nerve reaches the central nervous system with the signal that there is hypotension going inside our body, central nervous system respond by stimulating sympathetic outflow and vasomotor center in medulla oblongata. Okay, and the CNS is responsible for firing the sympathetic outflow, and you know that sympathetic outflow is responsible for the vasoconstriction of the arterioles and the venules, and when there is vasoconstriction of arterioles and venules, the blood pressure is going to rise, right? So that's what we need the blood pressure to rise in case of hypotension. So what are the sympathetic fibers and juxtaglomerular apparatus? Sympathetic fibers are triggered in response to the glossopharyngeal nerve coming with the signal that there is low blood flow towards the carotid bodies. So blood pressure drops. It's the first scenario. Vasomotor center has been activated. It increases the sympathetic outflow by the vasomotor center in the medulla, and these vasomotor centers release norepinephrine's, right? They are postganglionic and preganglionic nerve fibers, and the postganglionic nerve fibers originating from the sympathetic outflow, they releases the norepinephrine. Norepinephrine, it's the very important stimulant. It reaches the juxtaglomerular apparatus, and this apparatus has beta 1 adrenergic receptors. What does it mean? It means that beta 1 adrenergic receptors present in the juxtaglomerular apparatus, these are sensitive to the norepinephrine. These are just receptors for norepinephrine. It means that they respond to the norepinephrine and they are get triggered. It result in the release of more renin, and that's what we need initially, right? So let's talk it in case of diagram. The carotid sinus in case of hypotension, action potential via the glossopharyngeal nerve, the vasomotor center gets activated, okay, and there will be sympathetic outflow. Sympathetic outflow, the preganglionic fibers, the postganglionic fibers, and these postganglionic fibers releases norepinephrine, and this norepinephrine is going to be acting on beta 1 adrenergic receptors in the juxtaglomerular cells, and in response to the norepinephrine, these cells are get activated and they release the renin. Medications are of very importance. Medications such as beta blockers. Beta blockers means that those medications that are going to block the beta 1 adrenergic receptors that were responding to norepinephrine. It means that the beta blockers inhibit these beta 1 adrenergic receptors to treat hypertension. Because in case of hypertension, when there is already blood pressure increased, we do not need renin. We do not need vasoconstriction. We do not need sympathetic outflow, right? So beta blockers, they block the norepinephrine receptors and to treat the hypertension. Exactly. What are the functions of renin? The renin is released in response to the pressure reduction, in response to the sodium reduction reaching the macula densa, in response to the sympathetic outflow, in response to the norepinephrine beta 1 adrenergic receptors, right? And renin enters the systemic circulation via the renal vein. Liver produce a lot of proteins, as you know that many of the proteins are produced in the liver like albumins, globulins, fibrinogen, etc. except the gamma globulins. Gamma globulins are immunoglobulins produced by the plasma cells, right? So Angiotensinogen is a precursor form of angiotensin. It is also produced in the liver. Let's talk about the angiotensinogen converting cascade. What does it mean? Renin has already been released by the renal vein, right? So renin is going to act on angiotensinogen. It is going to convert angiotensinogen to angiotensin 1. Angiotensin 1 is going to go through the pulmonary circulation. There it will going to be converted into angiotensin 2 by the angiotensin converting enzyme ACE, right? And for more information, the angiotensin 1 is decapeptide. It contains 10 amino acids, and angiotensin 2 is octapeptide. It contains 8 amino acids. To be more specific, the angiotensin converting enzyme is not present inside the lungs. It is not present in the alveoli, it is not present in the interstitium, it is present on the surface of the pulmonary capillaries, endothelial cells, right? Angiotensin converting enzyme is an ectoenzyme. It means that it is present outside the lungs, to be more specific, on the pulmonary capillaries endothelial cells. So let's see this diagram. In this diagram, the angiotensin 1 is coming. It is been converting into angiotensin 2 by the action of angiotensin converting enzyme, which is present on the surface of pulmonary capillary endothelial cells, right? So this is the function of angiotensin converting enzyme. Another function of angiotensin converting enzyme, it inhibits the production of bradykinin. And it makes sense because bradykinin is a vasodilator. It relaxes the blood vessels. So in case of hypotension, when there is already blood pressure reduction, we are not going to use the bradykinin. So angiotensin converting enzyme is very smart. It converts angiotensin 1 to angiotensin 2, which is a vasoconstrictor, and simultaneously it blocks or inhibits the production of bradykinin, which is a vasodilator. We do not need vasodilator in case of hypotension. So the balance between vasodilation and vasoconstriction maintains the blood pressure. And when, of course, vice versa, when there is increased blood pressure, so it will going to release the bradykinin, right? So there are medications, again, angiotensin converting enzyme inhibitors. It means that they block the function of angiotensin converting enzyme. Okay, and these drugs includes captopril, enalapril, and lisinopril. Okay, these are very important angiotensin converting enzyme inhibitors. They do not allow the conversion of angiotensin 1 to angiotensin 2, and they do not allow the production of aldosterone. Okay, in case of hypertension, again, these drugs are used to treat hypertension, not hypotension. This is the overall diagram representing that how the system renin angiotensin aldosterone system is going to work. So let's repeat. First of all, when there is reduced renal perfusion, the blood flow towards the kidney is reduced. The juxtaglomerular cells are triggered, they release the renin. Second, the chemoreceptors which are present in the macula densa at the junction of thick ascending limb and the distal convoluted tubule are triggered in response to the low sodium reaching there. They are also triggered to release the renin. Third, carotid bodies. In response to the hypotension, they trigger the CNS by glossopharyngeal nerve. Their sympathetic outflow is fired. The postganglionic sympathetic fibers are going to go towards the juxtaglomerular cells, and these cells have beta-1 adrenergic receptors. In response to norepinephrine, they are also going to release the renin. The renin has been released. Angiotensinogen by the liver is converted to angiotensin 1 by the renin. Angiotensin 1 is going to go through the pulmonary circulation. There it's going to convert into angiotensin 2, a vasoconstrictor, by the enzyme known as angiotensin converting enzyme. And this angiotensin 2 has a lot of functions in maintaining the blood pressure, in maintaining the hypotension. There it's going to release the aldosterone, and aldosterone, the main function is to retention of water and sodium in the distal part of nephron. So the blood volume is going to rise, right? And vasoconstriction, it will cause the constriction of arterioles and venules. Again, the systolic and diastolic pressure is going to rise. That we're going to discuss in the next lecture. So this was for today. I hope you understand the basic concept regarding the RAAS, and I will see you in the next video.